Today we're going to have a discussion about the different measurements of disease, and this is more of a mathematical lecture, but it's also combined with some very important concepts. So first of all, when we observe a disease in the population, and the kinds of examples we're going to discuss are very acute diseases that occur very quickly. These are infectious diseases and they involve microorganisms. Generally, the types of disease and the types of organisms are found in food. So we hear often on the news about various foods being pulled off the market because they're contaminated with some kind of microorganism. So today's lecture is going to be about learning how to figure out is a disease actually occurring, number one. What is the extent of the disease? Is the disease affecting a lot of people or only a few? Who's at risk for the disease? Is the disease more likely to occur among individuals who are elderly, who are young, who, a certain gender, certain income level, certain racial group? Are the individuals who are getting the disease located in a certain geographic area? So all of these lines of evidence helps us determine indeed, A, what is the disease and what is the source of contamination. The other thing that we need to know is how is the disease transmitted? Is it transmitted by the air, by the water, by blood products, by an insect, by an animal, and also is it carried by food? How is the disease defined? What kind of symptoms are we seeing among the individuals who are ill? Are these symptoms held in common? Do we see variation? All of this information is truly important in relationship to figuring out, what is the source of the disease and more importantly, what can we do to eradicate it? So one of the measures, the first measure we're going to discuss is getting a count. Now, that's obviously a pretty simple thing to do, right? To count the number of individuals who develop the disease. But while the assessment of the extent using the count measurement is a rather simplified approach, what isn't simplified is thinking about the underlying population. Who is the population at risk and how numerous is that population? And that will certainly make a difference in relationship to how many, for instance, of a particular disease, of how many individuals actually develop the disease. So here is the first example. So you can see here, this is a count of tuberculosis cases starting in 1980. And tuberculosis, the incidence and prevalence of tuberculosis was on the decline until the HIV, the outbreak of HIV, and individuals who have HIV are very susceptible to developing tuberculosis. So once, year or two after the, introduction of the AIDS virus and the beginning of the AIDS epidemic, we also started seeing an increase in the expected number of cases of tuberculosis. So that was a really important observation that then led individuals, investigators to consider that individuals with HIV and AIDS are at high risk for developing tuberculosis. Now this example is an important one as well. So one of the things that we discussed in the other lecture was constructing what's called an epidemic curve, where you actually count up the numbers of individuals who have the disease, in this case on a daily basis, on an hourly basis, or in the case of influenza, on a weekly basis. And then you can see that, and we know, for instance, with influenza, that there are certain times of the year where more people develop the flu. Of course, that time being, it's about October, November through till the middle of the spring. But the important thing here with this particular epidemic curve is the new H1N1 influenza virus that came on the scene in 2008 and 9. The important thing that was noticed right away is that particular strain of influenza occurred at a time when at a time where the normal occurrence of influenza didn't happen. So it was really in this outer time period that we actually saw H1N1, and that was initially the way that individuals could determine, for instance, that indeed they had H1N1 and not the normal flu. So if you were around that time, and if you happen to be in a high-risk group, or if you were a healthcare provider, basically we all went out and got vaccines at that point, because it was a pretty serious epidemic that the H1N1 was a brand new influenza virus that had never been experienced by the human population. So in any event, developing that count was really important because we could see right away that H1N1 occurred at a different time. Now, here's another example. So here are the counts of AIDS cases, and this is cumulative through 2007. And you can see that the AIDS cases are clustering in populous cities. So we have Seattle, San Francisco, Los Angeles, Miami, New York, Chicago, Houston, and Dallas. And so there are two things to think about when you see the distribution of a disease like this. Number one, what about the population at risk? So we know, for instance, Los Angeles, San Francisco, Miami, and New York have high numbers of people who are in the gay community. And men who have sex with men are the highest risk group for HIV/AIDS. So it would make sense that the clustering would occur in those areas. But the second point to note with that also is that these areas have a very large population base. So if you're only relying on counts, for instance, and you don't think about the denominator, you might think that having such a high count was completely due to the numbers of people who are part of the gay community, for instance. But you know, we also have to see that those areas also have a very large population base. So another example of that is the prevalence of HIV in San Francisco is much higher than it is in Los Angeles, but the numbers of AIDS cases in Los Angeles is much more numerous than in San Francisco. And it's really because Los Angeles has a much larger population base, and so therefore they would also have more cases, but the prevalence is much lower in Los Angeles than it is in San Francisco. So let's talk about a food outbreak investigation. And this is the rest of the lecture is going to discuss the particulars of the different steps and tools and techniques that you need to have in order to conduct a food outbreak investigation. So the steps involved in determining indeed that a food outbreak has occurred are very prescribed. They're recommended by the Center for Disease Control. We're going to talk about those in depth. Also, but it isn't just determining the cause, right, of the illness. It's also determining the source, because ultimately, for instance, if it's a particular food item, then if we pull the food item off the market, then that's an immediate, cause and effect relationship. So if the cases go down, then we know we found the source. But if the cases don't go down, then we have to keep looking. So here's an example, a large supermarket with plenty of different types of fruits and vegetables, and perhaps there's a parasite or a pathogen living on strawberries, and we need to find out if people who are coming down with a particular illness, whether or not it's the strawberries that were the culprit. And as we become more automated and international in relationship to the types of foods that we eat, these steps have become more complex. So here's a picture of the potential sources of contamination that could occur in relationship to a food outbreak. So we have, if it's seafood, of course, we have the boats. If it's meat or milk or dairy, we have cows. We also have chickens, right? We've had contaminated eggs. The environment in which those foods are produced. So in the case of farms, the soil, right? The contamination has occurred by growing things in contaminated soil, for instance. The storage, the barns, the processing plants in terms of meat and other foods, distribution, where and from which locations did the food come from? How were they distributed? Then the different retail establishments, were the foods located in a particular store or group of stores, or were they widespread all over the United States? Were these foods consumed in particular restaurants? And the example we're going to look at is a restaurant example. What about the preparation methods? So is it because the food preparers didn't wash their hands? You could have contamination in the home. So if you bring contaminated food to the home, then you may, or there may be leaving a food out for too long before cooking. That could be a source of contamination as well. Then we have our human consumers, right? And perhaps only a certain segment of the population is experiencing the disease. We have restaurant customers, and then we have things like salad bars where everything's all mixed up, and then it can be very challenging to figure out which food is responsible. So there are a series, as I described, there are a series of prescribed steps that generally are done when, for instance, the CDC is involved with the investigation. So the first step is really determining, indeed, did an outbreak or is an outbreak occurring? And it's then, defining and outlining what constitutes a person who is ill, what signs and symptoms do they have. And then after you collect that preliminary data, you begin to generate hypotheses. And those are questions that you ask, and then you go out and you collect data in order to address the questions. So in generating these hypotheses, you obviously want to make them specific to the types of information that you are able to collect. Then, after you collect the data, you actually test the hypotheses, and you test them through analytical studies, laboratory studies. You assess the food itself. You see if pathogens are in the food. You look at various specimens of the individuals who are ill. And, if the common denominator happens to be a particular strain of bacteria or a particular virus, then at least at that point you've established the pathogen, and that's really important. Now, if, for instance, at that point you pull the food off the market and you didn't find that it made a difference, okay, then you have to repeat. You have to start, if the cases continue, you have to go ahead and generate a new hypothesis. And this actually occurred in the Salmonella St. Paul example that I described in the other food outbreak lecture. So then, for instance, after trying another possibility, and if then indeed you find that you found the food, right, you found the pathogen, then you can conduct a intervention, right? You can take the food off the market, and then the cases will go away. So, but, in today's world, it doesn't just stop there. The last part of the piece is determining the source. Where did the contamination come from? Because if you don't determine the source, then potentially it could occur again. And that part of the investigation can be extremely challenging. So then when all the cases are gone, right, then you can declare that indeed the outbreak is over. So the example we're going to discuss is a food outbreak that occurred in Michigan in 2006 in the early winter months, January and February. And what happened was this local health department was notified that there was gastrointestinal illness in members of dining parties that had eaten at a particular restaurant in the county on a very particular day, January 28th. So the health department initiated an investigation to try and identify the source and the agent of infection, and to figure out how many patrons of the restaurant or employees actually were taken ill by this particular event. So conducting the investigation revealed that several food service workers had become ill during that period of time. And in particular, a line cook had thrown up in the restaurant. Isn't that just wonderful? We don't really know what goes on in the kitchen, do we? And certainly that was a potential way that the environment became contaminated, and potentially the way that the virus was transmitted. So the outbreak investigation determined that 364 patrons had become ill from this particular event. Now, it was determined that the pathogen was the norovirus, and this is very relevant because the contamination that has occurred recently in all of these cruise ships, by and large, have been because of the norovirus. So this is something that is going on in today's time, and you'll note that when they describe the outbreak, it's very severe. It infects persons of all ages, and there's a particular peak month or peak time during the cold winter months. Also that the virus is transmitted primarily by the fecal-oral route, and experiencing the infection will cause extreme vomiting and diarrhea, which if you pay attention to the news reports, that's exactly what they describe in these cruise ships. And with the cruise ship example, these people are basically locked into that environment, and it can be something very minimal, just a handle on a door. If one person gets it, or if the point source is the kitchen, food service worker, then potentially the whole ship can become ill. And as we've seen in the news, it can be really dramatic. So in terms of recognizing an outbreak of norovirus, there's a short incubation period, just 24 to 48 hours. There's a very short short period of time that individuals are ill. There's a high frequency of vomiting, and there's an absence of bacteria. So that's important. So if there's an absence of bacteria, then you would assume it's a gastrointestinal virus, and norovirus is getting to be the number one. So anyway, so this is a legitimate guess on the part of the investigators. And so what enables, for instance, that guess is observing the symptoms, right? And taking specimens of the individuals and then seeing indeed it's the norovirus causing it. However, you still don't know what the food is, right? And if it's a nationwide outbreak, that becomes very important. So here were the cases of norovirus over time. So you can see it's over this very short period of time. And once the interventions were implemented, then the number of cases went way down. So then if you implement a particular intervention, the cases go way down. Then you found your agent. And then you can see the incubation period. Well, here's the vomiting incident from the employee. And the incubation period is about correct, right? It's about 2 days, 1 to 2 days. And then you get this high number of individuals who came down with this illness. So let's talk about the numeric measurements that you need to do when you're conducting a food investigation. So the first one that investigators often do is called an attack rate. And that's where you simply look at the number of people who are ill, who've taken ill, and you divide them by the total number of people at risk. So, for instance, if we're doing an outbreak investigation in a cruise ship, right? The denominator would be people who are on the cruise. Okay, in the restaurant example, it's people who ate in the restaurant. Now, and we're going to see there's a slight variation on that theme in relationship to the denominator. But the denominator selection of the denominator is very important, because you don't want to choose people, for instance, that are eating in the restaurant down the road. You want to restrict the investigation, the analysis, to the people who truly are at risk of getting ill. And that becomes a challenge, again, of course, when you're talking about nationwide outbreaks, because then it's very challenging even to figure out who the population is at risk. So often, for instance, if they pull food off the market, they'll give lot numbers and so forth. But you know, many of us don't have time to look at the lot numbers, and I would suspect that there's a lot more cases of food-borne illnesses than are reported, but certainly the serious cases do get reported. So in the case of our restaurant example, we had a total of 281 individuals that had dined on that particular January 28th, and that computation resulted in an attack rate of 33.7%. Now, the next day, the attack rate actually went down, which suggests that either there was an intervention, or, you know, the word got out and people heard that the particular restaurant, you know, there was a food outbreak, so they stopped going, or something, right? So we could see, so the attack rate is important because it tells us about the extent of the problem. Now, we don't know anything about the food, right? But we do know about how many people are ill, and we know the proportion. We have that denominator, which, as we could see in the examples also, is very important. So in terms of computing the attack rate, now remember, 281 was the number of people who became ill. Now, what they did in the restaurant is, instead of using the denominator of people who actually were visited the restaurant, they looked at the number of meals, and we're going to see the relevance of that later on. And the number of meals in that case was actually high. It was 834, and if you divide the two, you get 33.7% attack rate. And then, of course, the next day, the attack rate went down. So here is looking at the attack rate over these very prescribed intervals. And in this case, it was intervals of time, you know, it was hours, right? So 3-hour intervals. And so the peak occurred between 5 and 8 p.m. on the 28th. And then we saw on the 29th, the attack rates went down. So, you know, but we did see elevated attack rates during the, you know, lunch hour as well. So this helps us in relationship to the food too, you know, what foods are eaten in common both at lunch and dinner. So this is a description of the types, the characteristics of the people who became ill. They were, you know, median age was 40. They were mostly female. They had a quite a high rate of diarrhea as the symptom. And some of the individuals had to seek medical attention, 8%, and some went on to visit an emergency department. So, you know, the mean incubation period was about reflective of the norovirus. Duration of illness was the same. So sometimes just knowing the characteristics of the people who become ill helps you figure out what pathogen is responsible. So the next kind of analysis that you do is called a case control analysis, and that helps you determine which particular food is the source of the pathogen. So we're going to show you, we'll see directly how an odds ratio is computed, and then we'll actually try one out in our example. So there were two types of food that were evaluated, the antipasti and the garlic. and the antipasti had an odds ratio of about 3, and the mashed potatoes was about 4. So we'll see the relevance of that. Now, odds ratio that basically demonstrates no association. So when an odds ratio is equal to 1, that represents no association. So what you're looking for in a food outbreak is to find foods where the odds ratio is greater than 1, and the confidence interval also doesn't cross 1, which means that it's a significant association. So in most outbreaks, you actually look for odds ratio that are much bigger than 1. So the example, the Salmonella St. Paul example, we saw some odds ratio there, I think of 60 or so, which suggests that's a huge odds ratio. So the particular food or foods that were identified in that case were very strongly related to the pathogen. So then what you do here is actually, this is the case control analysis, not the attack rate. And you actually create a cross product, and that's what produces the odds ratio. And again, in the example, we're going to go through that in detail. And the food outbreak measures then are both the attack rate, and we do the attack rate to tell us the extent of the disease and how serious, what the numbers are who were exposed and the numbers in relationship who developed the disease. So when we have a really high attack rate, then we're pretty clear that A, it's a serious infection. And, you can go back to the infection patterns lecture, and, there's a high dosage, right, of the virus. If a lot of people get sick from eating the food, then potentially the amount of pathogen, in this case, the amount of virus in those particular food items could potentially be high. So as I mentioned, the important part of it is really figuring out the appropriate denominator. And, in the case of some analyses, it's the total people eating at the restaurant. And the case of the norovirus, they used the total meals consumed. So then you compute the attack rate during a certain period of time. So that's important because obviously with a virus like norovirus that has a very short incubation period and a very short time that people have symptoms, then the denominator and then actually the assessment of the attack rate has to occur during very narrow time periods. And by doing that, you can help determine the time and the place of infection. So you could do food outbreak measurements by place, by restaurant, by institution, by market. So how you do it is really important because it can give you a lot of clues in terms of what's going on. So then after you have your attack rates and you see that indeed it's a problem, then you construct possible hypotheses or questions. And you construct these hypotheses by examining the evidence that you already have and looking at your data. And also, but also very important is your knowledge about certain symptoms and signs related to particular pathogens. So in the case of the restaurant, you might say, well, we suspect that the norovirus is responsible for this particular outbreak, and we are going to assume that, and if we get information about the food service worker, the cook, right, and you can say, well, gosh, I unfortunately vomited near the garlic mashed potatoes or the antipasti, right, then that would help you ask the question, is it one of those foods that was responsible for carrying the norovirus or that was responsible for harboring the norovirus and was responsible for causing that norovirus infection. So in order to do that, you conduct the case control analysis and you evaluate particular foods in relationship to the other foods. So the first example is exactly that. We're actually going to compute an odds ratio by hand. Now, generally, you have models and analyses, right? You don't usually need to do them by hand, but, you know, if you ever get an MPH or a PhD in public health, and you will be asked to do one of these, I guarantee it. So anyway, so let's get our table back. So here are the ill individuals, and here are the ones that are well. Okay, and then we have our antipasti. Okay, our garlic. And our garlic mashed. Okay, and then our other foods. Okay, so we had 10 individuals antipasta who were ill, 10 garlic mashed, and 25 other. Okay, and then our well, we had 8 and 6 and then 77. Okay, so when you have data like this, what you need to do is you need to construct a 2x2 table. And in the 2x2 table, you'll have the ill individuals and the well individuals. And you set up your cells, right? And this cell here is going to be what's called the exposed individuals and the unexposed. And then you label your cells. You always need to set up the table exactly this way, because if you don't, then your calculations won't work. So you label this cell A, B, C, and D, and then the odds ratios formula is A, or here I'll make it lowercase, A multiplied by the number in D divided by B multiplied by the number of individuals in C. So in each of these cells are counts of individuals. So if you set up the table exactly like this and you use this calculation, you will be able to compute your odds ratio. So if we take the data from the restaurant example and fill it in these cells, so we have our, so let's take, because we didn't compute that, we didn't show that in the computation, let's take garlic mashed. So we'll have garlic mashed here, and then all other foods in the unexposed category. So we have 10 individuals who were ill and 6 who were well. And then to compute all the other foods, we have to add them up. So the ill individuals, remember these are ill and these are well. Okay, so that's 10 plus 25. And the other cell is 8, okay, plus 77. Okay, plus 77. So that works out to be, and I'm always somebody, I do everything step by step, right? 10, 6, 35, and 85, right? Okay, so there you go. So then you do the computation. So okay, so I guess, well, let's go this way. So we could do 10, right? A times D, which is 85, okay, divided by 6 by 30 times 35, right, just by using the AD over BC. And if you work that out, guess what? It equals to 4.05, which was our odds ratio for the garlic mashed potatoes. Now, again, you look for the foods that produce the large odds ratios, because then they're very likely to harbor the pathogen. Okay, now in the case of the restaurant, right, you can just, if it's a food service worker, for instance, you can treat them, have them not work in the restaurant anymore, and then take care of the outbreak very easily. But in the case of a larger outbreak, or an outbreak that happens with food sold in a market, then you have to take all the food off the shelf. Now you can do that, and if you still have cases, then you have to go back and rethink your hypothesis. So all of these steps are pretty prescribed. They need to be, because they have extreme relevance in relationship to a lot of people dying and getting ill, and it's just not a good thing. So it's important that the people conducting these investigations are very thorough and very complete in terms of what they do. So let's go on to the second example. So the second example is, why do you think the norovirus example used meals consumed rather than people as the denominator? Well, guess what? Sometimes you can't find all the people. So, ascertainment, okay. Finding people who ate at the restaurant. What if they paid in cash, right? If they paid in cash, you have no record, all right? And you also, unless you kept your receipts, you don't know also who ate the food, right? Now, the great thing about these debit cards and these credit cards is you have an electronic record then that this individual indeed ate at the restaurant. The other thing going on with this investigation was the time period, right? The time period was very short, and it was important, right, that the restaurant make this determination as soon as possible. So in the case of the restaurant, it was much easier for them to actually pull all their slips, from where the waitresses made their orders, and they could actually count up the number of meals that were served during these very short time intervals. So really, again, it brings up the important point about doing a good job about choosing your denominators. So being very creative sometimes and very thorough and rigorous in finding that true denominator that really reflects the population at risk. So the last example are, what are some of the sources of information used to analyze national level outbreaks? Well, I tell you, this is fascinating. A couple of years ago, I went to a national epidemiology meeting, and I sat in on a Food Outbreak Investigation session, and it was fascinating because they had people there from the health department, but then they also had representatives from some of these markets. And they were talking about some of the ways that they actually are able to determine the sources of food, the purchasing habits of people, and it's all now electronically traceable. So we all take our receipts from our, supermarket shopping experience and, toss them in the bag, right? But they actually have quite a bit of information on those things. And so this session, they were talking about all the different kinds of information they can collect from those receipts, those, paper roll receipts. And they can, first of all, determine, what kind of fruit or what kind of vegetable, where did it come from, who was the distributor, whether they can get back to the farm is another question, but that's really what the food safety industry is really working towards, to have that deep and comprehensive a level of ascertainment, because basically as our food distribution and our food purchasing and as the sources of our food increase to become international, we didn't have, we had a fishing boat there. I guess we need to have shipping containers too, right? That as it becomes more complex, it becomes more and more necessary for us to develop these very high-powered, analytically sound, large, big data, right? Big data is involved in this, and it's very important to be able to conduct these complex analyses. Now, what we did here was very simplified, but in a nationwide outbreak, it's much more complicated. So, thank you so much for visiting educator.com. This concludes our lecture for the day.